everybody. So today we're going to learn about regulation of gene expression, cell specialization, and mutations. So gene expression just means the amount of proteins that are being made, and your cells have ways to regulate that. And so here's just a few definitions of vocab for proteins and words that are going to be important as we go through this lecture. So gene expression is the process where genes in your DNA are transcribed and translated. So basically that just means protein synthesis. And you've got different chemicals that are going to regulate this. So you have regulatory sequences. Those are part of the DNA that can either promote or inhibit transcription. You've got proteins that interact with the regulatory sequence that are going to either inhibit or activate. So if you look at this picture, here is your region of your DNA, and what happens is it folds. So you have your enhancers, and when transcription factors bind to them, this whole thing is going to fold and basically activate this part, which is the promoter. And the promoter, remember, is where RNA polymerase is going to bind to. So all of this has to happen in order for transcription to occur. So eukaryotic cells, we know, are more complicated, and most of gene expression is controlled in transcription. So that would be all of the processing. Remember, the introns being spliced out and the exons being put together, the cap and the tail, all that stuff is going to be how the eukaryotes control gene expression. There are some things that happen after, but the majority is in transcription. So epigenetics is a topic that is really new to the genetic field in the last 15 or 20 years. And what it involves is reversible chemical modifications that happen to your DNA as you live. So the choices that you are making now in terms of your diet, in terms of your environmental, like if you smoke or if you have a lot of stress or those kinds of things, actually can affect your genes. It doesn't change your DNA, but it changes how it's coiled. And that can actually be passed on to your kids. So just know that the choices that you make now in terms of how you treat your body actually can be passed on to your kids. So what's happening is, if you look at this picture, and I put this in words right here, so if you want to write them down, you can. But if you look at this picture, remember that histones are what DNA wraps around. So this whole thing is called a nucleosome. And we know that in order for RNA polymerase to bind to your DNA and transcribe a gene, it has to be available. So something like this is where your DNA is wound up more loosely and the gene is available for transcription, so that means that protein can be made. If your DNA is wound up too tightly, like this gene right here, it's not accessible to RNA polymerase and it's basically turned off. And so chemicals can either wind DNA up tighter in regions or it can loosen it. And so that basically turns off the gene or turns on the gene. Some other things, so these are just a summary of how gene expression is controlled in eukaryotes. So the epigenetics, so activating and inactivating. We talked about introns and how you can remove them and remove different ones with alternative splicing. And then also the modification, like the cap and the tail, and how it can get through the pores. So remember, the longer the poly A tail, the longer the messenger RNA will stay in the cytoplasm and work, because it's not getting broken down as quickly. So also, some things that always come up is, well, so all of your DNA is in each of your cells. So how does a muscle cell know to be a muscle cell versus an intestinal cell know to be an intestinal cell? So what happens is there are specific proteins that are made in each of these differentiated cells that then causes them to become more differentiated. And so the presence of specific proteins within cells of a tissue, and tissues are groups of cells that are working together, give the tissue its function. And so the gene expression, what proteins are made, causes cells to become their different types. Okay, so operons. So here we're going to talk about mostly prokaryotes, but some more language on control of genes and expression. So operons are closely linked genes that make a single mRNA, and the one that we're going to talk about most is what's called the lac operon, and that's in prokaryotes in E. coli. So this is an example of an operon, this whole thing. So you've got regulatory genes, you've got the operator, and then you've got the genes that you're going to transcribe. So the operator, remember, is going to be there to either inhibit or promote transcription by binding with a regulatory protein. So let's see how this works. So it's what's called inducible, and it's inducible because it's normally off. So lac operon is a set of genes that makes lactase, right? So to break down lactose. So the only time that this lac operon is working is when lactose is present. So when lactose is absent, which is normally, your protein that was made from your regulatory gene is going to bind to the operator and block RNA polymerase from transcribing the gene, right? And we want this because you don't want to be making all of this protein if you have no lactose to break down. So this is when lactose is absent. I would go ahead and make a drawing of this in your notes, or go back to, if you've drawn this before, make sure you've got all the parts. So when lactose is present, then it turns on and makes your protein. So inducers are molecules that bind to this repressor and pull it off of the operator. So in this case, allolactose is the inducer, and it's just a type of lactose. So when there's a lot of that around, it's going to bind onto the repressor and pull it off, which allows RNA polymerase to bind, and then it can go it can transcribe the genes and then make the protein that's going to eventually break down lactose. So that's when lactose is present. So there's another type of operon called a repressed operon. So here's the definition of the two. So you have an induced operon, which is always off until you induce it to turn on, and that's the lac operon. A repressed operon is opposite. It is always on until you repress it and stop it, and that's what's the trip operon. So trip is short for tryptophan. That's the amino acid that's in turkey that we talked about that makes you sleepy. And the trip operon is always on unless there's lots of tryptophan present. So if you just eaten a big turkey dinner, then maybe this operon will turn off. But here's just a picture. There's a lot more detail than you need to know. But here's your tryptophan, right? And it's binding to the repressor, and it's going to actually cause it to stop working, which is why this is different. It's repressing the making of tryptophan because there's a lot present already. Okay, so how do we get different phenotypes? So promoters, we talked about those, and that's in your green right here. Those are sequence of DNA, and that's where the RNA polymerase is going to bind. But if you notice, these activator proteins, which are these color right here, different ones are made in different cells. So this is a liver cell, and you have a liver regulatory gene and only liver activator proteins, and so this is going to make specific proteins that are important to liver cells. If it was an eye cell, right, then you would have a different regulatory gene and different transcription factors, and so you would transcribe different genes. Okay, so next thing we're going to talk about is mutations and how they can change phenotypes because they change gene expression. So remember, mutations are changes in DNA, and they can either be beneficial, harmful, or most likely they're neutral. And that all depends on the effect of the environment and natural selection, which we'll talk about in our next unit. So mutations are also a primary source of variation. So they are totally random You don't try to mutate your DNA so that you can survive better. It just happens and you get lucky. So there's a couple we're going to talk about. Base pair substitution, which an example is sickle cell anemia. And if you notice, here's your DNA and here's the correct protein, right? But with sickle cell anemia, it changes one letter and it changes this amino acid. Instead of glycine, it changes it to cytosine, and that in turn changes the whole structure of hemoglobin. So that is what's called a base pair substitution. And then the next one is an insertion or deletion. And these are often really serious because they can change the order of the sequence after your insertion or deletion. So remember, DNA and messenger RNA is read in threes and codons. So if you insert mRNA, if you insert an mRNA, like an extra letter there, and then your mRNA is going to have an extra nucleotide, then everything after that is going to be messed up, and you're going to change your whole amino acid sequence. The other thing that can happen is, remember, stop codons. If you change the DNA, if you insert a letter, it could actually cause a stop codon to happen, and then the protein doesn't get made. Now, those were genetic mutations. These are chromosomal mutations, which we talked about in meiosis. So remember non-disjunction, where the chromosomes don't separate correctly, and you can get a triploidy, which is 3 of 1 chromosome, like Down syndrome. And that would be trisomy 21, and the way that you write it is this. So that would be a girl, a female, and it would have 47 chromosomes, and that includes the extra 21. Turner's, remember, is where a female only has one X, so she only has 45 chromosomes, and she's missing an X chromosome. The other interesting thing that can happen is what's called polyploidy, and that happens in plants often, and that's where you have 3 or more of every chromosome. Now, this does not happen in animals, they would miscarry, but in plants, it definitely can cause them to become a new species. Some other ones, I think pictures do this justice, so if you want to draw some quick pictures. Inversion is where 2 chromosomes just change, they flip over, like you invert it, you flip it. Translocation, what's happening is this part of the chromosome is just getting stuck onto another non-homologous chromosome, so they exchange segments. And deletion is where a whole section of DNA just breaks off. And then this one, duplication, is where you would have a chunk of a chromosome that would double. Okay, and then in prokaryotes, so that was all eukaryotes. In prokaryotes, you get increased variability with what's called horizontal transfer of genetic information. So transformation, we're going to do a lab on this, is where a bacteria would take up naked DNA, which means it's not in a cell, just from the environment. And what we're going to do is we're going to have E. coli take up DNA that's going to make them antibiotic resistant and glow in the dark. Transduction is where you have a bacteriophage, so this is a virus, and it's going to insert its DNA into a bacteria and give it just more different DNA. And then conjugation is where one bacterial cell hooks up with another one, and for example, like this little piece could travel through this connection into the other cell. And that's it. So make sure you have some good drawings, took some good notes, go back, and hope you learned something.